Hello, everyone. Thank you for joining us today. My name is Jay Singh, and I will be your host for this NASA Technology Transfer Program webinar on the various health, medicine, and biotechnology technologies from NASA Ames Research Center. The webinar will be structured in a lightning talk format, so the presenter will have 10 minutes to present their technologies, then 5 minutes for Q&A. Before we get started, I wanted to mention the Technology Transfer Program has over 1,000 patented technologies across the entire agencies and 100 different technologies at Ames available for public to license. In grouping our health, medicine, and biotechnologies together, hopefully you'll come away with understanding of several items that are related. I'd also like to point out that your microphones will be muted throughout this presentation. So if you have any questions, please type them in the Q&A box. We'll answer them during the Q&A session at the end of each presentation. So at this point, I'm going to share my slides, and our first presenter will be presenting. And our first presenter is John Galeska. John, take it away.
Thank you very much, Jay. So I'm going to quickly tell you about an invention called methyllotrophic microorganisms expressing soluble methane monooxygenase proteins. Next slide, please. And I'm going to begin, now, while this invention has broad applicability to the biotechnology industry, I'm going to begin by describing our motivation. And I think that's best described in the image in the top right there. So on the International Space Station, presently, the CO2 removal system relies on a technology called Sabatier. And Sabatier reacts excess carbon dioxide from respiring astronauts with hydrogen to generate methane. And you can see that CH4 indicating methane generated by that Sabatier unit. Now, presently, that methane is exhausted off of the ISS. It's not captured or reused. And our motivation was to find a way to utilize that methane as a carbon source for an in-space biomanufacturing process. And while there are multiple natural methanotrophs which can use methane natively as a carbon source, these bacterial species are not generally used in the biotechnology industry. They do not have robust genetic tools and thus have significant limitations. So our motivation for this invention was to find a way to modify the widely used biotechnology platform Pichia pastoris, which is a methylotrophic yeast, meaning it can naturally use methanol as a carbon source, so that it could now use methane. And looking at that left image, our invention is summarized neatly. What we describe is a way to express methane monooxygenases, or MMOs, from bacterial species within Pichia pastoris to allow Pichia to now use methane as a carbon source. Now, this invention is directly applicable to our goals in that it now would allow the utilization of methane produced by Sabatier in spacecraft, or eventually in destinations like Mars, for an in-space manufacturing process based upon Pichia pastoris. Pichia pastoris is appealing for a number of reasons. It's, again, widely used in biotechnology to produce protein products. This includes one of the fastest growing classes of therapeutics called biologics, so protein therapeutics. And so, again, the applicability, we see this as being directly applicable to the emerging space industry. This would allow us to use this technology for habitat construction, astronaut nutrition, and biomedical applications. But I also wanted to highlight the fact that this is applicable to terrestrial biotechnology. Methane is the major component of natural gas, in which there is already a robust infrastructure for the distribution. Methane is also outgasses from sewage treatment plants and landfills, and is actively captured from these locations. And so there's an opportunity here for biotechnology, terrestrial biotechnology, to use this invention to potentially improve the economics of their processes by using alternative carbon sources. So again, moving to benefits, we see broadly this as a step towards an in-space biomanufacturing process, and that would allow us to do a number of things in space that are not presently possible, focusing on the products that microorganisms are very good at making, in particular protein products, which really cannot be produced in any other chemical process. Another benefit, these carbon sources, so methane, there is a robust process for making methane already from carbon dioxide. This is the Sabatier process, and we see that being deployed potentially broadly in space, in Mars and in spacecraft. But this can also, methane, there are emerging technologies for converting terrestrial CO2 to carbon sources for reuse, and this is a route that could become prevalent on Earth as well. Another benefit is that we already have a waste stream of methane on the ISS, and recapturing this could help us close the loop in space and improve the efficiencies of operations. And what's particularly nice about this technology is that it couples a number of robust technology platforms. Specifically, the Sabatier process, which has been running for many years, potentially decades, and is well understood, and also the Pichia-Pastoris platform, which is already widely used in biotechnology, the biotechnology industry. And so this one link brings these processes together into something that will hopefully allow for new terrestrial and in-space applications. And so with that, I think I have time to take some questions in the chat.
Yeah, thank you, John. That was great. So as people are typing up their questions, I just had one. So you're saying that this technology you can take kind of the methane and use it for biomanufacturing. Could you do that here on Earth? Like take the methane that, let's say, landfills, put out, capture that in whatever way you can, and then use that methane to do some biomanufacturing?
Exactly. Yeah, I think that's a great point, and I think that's one of the things that we're trying to highlight. While our direct motivation was to use this in space, as you point out, waste treatment, sewage treatment plants, landfills, and also the natural gas infrastructure provides a way to harness methane as an alternate carbon source for biotechnology. So yes, we think there's a direct application of this technology to the terrestrial sphere, and we hope that it can improve the economics of a number of processes.
Oh, that's really cool. Yeah. Thanks for explaining that. So we have one question here. Can we scale the yeast up to increase the absorption of methane?
Yeah, I think that there's this technology, while it describes this route, I think there is a lot of room for improving the productivity. So all these quantitative, we're sort of getting some quantitative questions like that right now. How productive is this? How much room for improvement is there? And this is, I think, this will be scalable. That's the nice thing about biology, is you can use effectively scaling your catalyst. This MMO scales with the growth of the organism. In addition to that, as individuals iterate on this technology, we should be able to improve the expression as well as the activity of these MMOs, the processes surrounding this delivery of methane to the yeast. Is also an opportunity for improvement. So I think with this sort of key invention, there's ample room for process improvements that are being alluded to.
Oh, great. And so there's two questions here. What is the productivity of the yeast for now? And are there any negative side effects, non-desired products that can possibly come out of this?
Well, negative side effects. I'm trying to think. I wouldn't say there's any, we haven't seen any negative side effects at this point. And the productivity values, we don't have great values for those yet. So this really is an initial description of this invention with some descriptions of implementation, which you can find in the patent application. And the negative side effects, I don't. This is, expressing a new protein is an additional metabolic burden upon the yeast. So how that balances out is still an active area of investigation. But we don't see any toxicity effects per se at this point.
Oh, that's great. I think that's it for the questions. So thank you, John. Appreciate it.
You're welcome. Thank you.
And just as a quick note for everyone who's listening right now, I'll be going over how to license these technologies that we're talking about today at the end of the PowerPoint. So if you stick around, I'm going to be going over in a very easy way to how you can find out how to license these technologies. So thanks again, John. Appreciate it. Next up is Travis Boone. Travis, take it away.
Thanks, Jay. So this patent was generated through our Wet Lab 2 project, where we make payloads for the International Space Station to do biological experiments, specifically RNA expression analysis. The co-inventors on this are Jimmy Jung from Ames and Macarena Pera, and also from Claremont Biosolutions, Mark Brown. So next slide, please. So I'm just going to zoom into each quadrant here of the quad chart so you can see it better, and I'll explain a bit more. So next, please. So this is based on an issued patent, and as I mentioned, it deals with RNA extraction and how to use PCR in real-time amplification to quantify the different genes or different RNAs. We developed a single-use disposable system with a portable form factor, which of course is good for field use in general. We have preloaded buffers and lyophilized or freeze-dried reagents, especially the PCR reagents, which include primers and probes that identify the specific gene of interest, and the polymerization enzymes as well. There are other RNA stabilizers in there and DNase additives as well that are described in the patent. And that's for basically specificity and to not have degradation of the sample or the RNA that was extracted. One of the things that's specific about this IP is the chemical formulation for the assay that does the RNA extraction is of low toxicity. So unlike most commercial products where you have alcohols or K-tropic salts, we don't have that, and we did that intentionally to make it easier and safer to run on board the space station. We also have the capability to store things long-term. We've done a lot of long-term studies at both ambient and 4C, but let's say a good ballpark is around a year of shelf life, and probably longer than that depending on the temperature. It takes about 2 hours to run the process. This is similar, PCR is what's used for a lot of COVID testing. So when you hear people say it's about a 2-hour turn, if you're, from start to finish without being in a queue to get samples in, that's similar here. About 30 minutes of that is hands-on time with sample preparation and setting up the reaction tubes for the PCR. And then 90 minutes is walk-away time that the thermocycling and reading is done. Next, please. So the devices that are involved, it's really a suite of devices. In the upper left there, the heart of it is the sample prep or preparation module, and that's where the RNA is extracted. In the top center, We have some devices that are used to debubble that RNA sample, since bubble rejection in microgravity is always an issue with all kinds of experiments. We then load that into a repeater pipette, and the tip of the pipette you see in the debubbler picture in the center there, it's already inserted in the plastic device that basically uses capillary action to wick the liquid down to where you want to draw it and keep any air gaps separate. In the bottom left, we pipette the sample into reaction tubes that have that lyophilized reagents already in them, and they go on a rotor. The reason for the rotor, it's about a 6-inch in diameter rotor. The reason for that is so we can use it as a mini centrifuge and run it just by spinning on the Corvus drill that's up on station already. So it's a nice lightweight way to do that. And then those tubes get inserted in the bottom right hand corner into a thermal cycler. This one is made by Cepheid, called the Smart Cycler. And after 90 minutes, we generate data, which is in the form of fluorescent signal that comes off of the various target genes that we were looking for, and we can quantify how many of each was there. Next, please. And one more, please. So the heart of it in the sample prep module is made by Claremont Biosolutions, and that's a commercial product. It's a bee beater, basically a lyser, and then a capture column. So RNA or DNA capture columns that they do. And that's really the business end of where the extraction is being done. And the manual valves and syringes are to be pushed by the user. You'll see that there's just a small battery pack on board up in the left-hand picture, and that powers the bee beating for the lysing. Next, please. So this says, in our case, this has been used on station. We've done about 4 different missions where it's been used, and 3 different crew members use it. Much more, about 13 have been trained, but you never know with the rotation, different flights and delays and launches and whatnot. But 3 crew members have done multiple sessions, 11 in total. You'll see Jeff Williams in the bottom there using the sample prep module. In the upper right, Kate Rubins, you can see one of those rotors with the tubes in it and the drill that it's spun on. And Ricky Arnold also has used the system, and his hand might be a little hard to see, is one of those debubblers that allows us to debubble the RNA. So system's been actively used and it's been working well. Next, please. So applications, obviously for us it's space research, gene expression. There's been also some use of it for microbial monitoring, not on station, but on the ground with the folks that do that on station. So that was another aspect that was being looked into. And then the other sort of obvious applications for any device like this. So whether it's diagnostics, disease monitoring, food quality. So there's sort of a standard list of things you might want to do with a field-ready sort of instrument like this, or suite of components. Next, please. So the benefits, obviously the size and field use of it, or portability, is big, and the fact that the reagents are preloaded. I mentioned the long-term storage. One important thing about it is that the target genes, or the primers and probes of chemistry, is user-defined. So those are made to make whatever panel is of interest to the user. Now, most commercial products are with fixed panels, and just to make it economical to make a lot of them and have a panel of different target genes. But in our case, we design it for whatever ones are of interest. I mentioned the operation time and the battery power on the sample prep module itself. The thermocycler runs off of normal 110 volt AC. And the important part, many of the claims deal with the reagents of the RNA extraction. And so the key there is about that they're low toxicity, no special handling required, and the formulation is a bit different than what you would see in commercial assays on the bench today. Next, please. So that's it. If there's any questions, I'm glad to take them.
Yeah, thank you. That was great. As people are typing in their questions, I had one. Can this be scaled down further, or is this kind of the size where it is? Like, let's say if a company looks at this, goes, "Hey, maybe we can kind of shrink it down a little bit further." Is that possible, or is it just kind of stationary where it's at?
Yes, definitely. Part of the form factor is to make it easy for anyone to use manually and to meet different constraints or criteria for flying it. But the amount of reagents that are needed or used can be quite small, um, smaller than the amounts that we move around to make sure we have enough to sort of macroscopically move around volumes of fluid. But devices like this can be miniaturized more, and as long as you have enough extracted RNA coming off or eluted RNA coming off to fill the PCR reaction tubes. In our case, where the lyophilized reagents are already ready and waiting there.
Ah, great. So we have one question here. Are there plans to adapt this technology to proteins?
Hmm. I don't think that's been requested. I could be wrong. We may have other group members and managers that have heard about projects like that. In the past, I've been in the microfluidics field for quite a while, about 30 years, and there have been some projects that we're looking to do protein analysis and sort of analogous to the way PCR is used to look at different genes. So there are definitely some complementary technologies right now, and there are some assays, already, of course, patented for how to do that for proteins. We have not been asked about that, but I would have to think about that and see if there's functions that are missing in our suite right now, or if it would be basically a relatively simple protocol change and reagent change. But I'm not sure about that.
And one more here. How does it compare to present day available similar to technologies that are already out there? So available similar technologies.
Yeah, I would say the key difference is, well, from our point of view, one of the key differences was the formulations being different than I believe what you'll see in normal claims in a patent. And that's partly about getting things to low toxicity. And I know, for some having been with several biotech companies, sometimes, building a bit of an IP wall with some claims and, that are different, and distinguishable from other claims that are out there for products that have been around a long time. So I guess that's one angle that if I was back in the startup world, I would be looking at. As far as the form factor of things, the suite of devices we use, of course, there's different designs you can use to achieve each of those steps, and some of them could be integrated together. And there are companies that, for instance, Claremont Biosolutions, that help miniaturize that process further to still do that same kind of extraction of the RNA. So there are products like that. I don't know if anyone uses I don't think anyone uses the assay formulations that we do for the extraction, but engineering-wise, there are products that can do that and can miniaturize things more, as we would do also if it was warranted for the space station. And in fact, we have started projects to do that, to basically be able to process more samples in parallel, or at least sequentially, and basically miniaturize how many different samples and sample prep operations are going on at the same time with one crew member.
Great. Yeah, thank you. That's very helpful information. And I think that's it for the question. So yeah, thanks, Travis. That was great.
Sure. Thanks for organizing.
Next up, we have Dr. Thomas Murphy. Tom, take it away.
Thanks, Jay. I'm Tom Murphy. I was at Ames from 2014 to 2016 as a postdoc, and that's when we did a lot of this work. Dr. Lee Bebout was my mentor. So today I'm going to be talking about synthetic trees for ultra-efficient algae cultivation. Next slide, please. Okay, so we're trying to grow algae for a number of reasons. One is for human life support in space, for CO2 consumption and oxygen production. Another application for algae growth is for renewable biofuel production, as well as high value products. So this figure shows the current challenges with conventional algae cultivation methods, namely open pond algae production. This plot shows as positive bars energy output from algae cultivation in terms of the energy contained in the biomass, and the negative bars show energy inputs to algae cultivation. So most algae cultivation methods require more energy in than you get out of them, which is a problem if you're trying to do something like biofuel cultivation. A couple call outs on this plot are the energy inputs for pond circulation. So this is just for moving all the water around the pond. Water is heavy, and it requires energy to keep that water moving, to circulate the algae, to give it access to light and gases. Dewatering, the yellow bar, is also a large energy consumer. Most open ponds, even when they look like a very dark green color, that's typically about 99% water by volume and about 1% algae. Next slide, please. So to address some of these issues with dewatering and pond circulation, this is our synthetic tree concept. So on the left, we have a leaf, like as an actual leaf as it exists in nature. So how a leaf works is as water evaporates from the underside of the leaf, fresh water, sap, or nutrient media is pulled up from the soil through the xylem and into the leaf to replace that water. So the cohesive nature of water provides a passive pumping system for the leaf. And in the center here is kind of our version of this concept. So at the top of the center figure, you see evaporation happening from this exposed region. So this is exposed to relatively dry air. And as water evaporates from that top region, fresh water nutrient media gets pulled in from this reservoir at the bottom. So the water is all moving through this this porous wicking membrane, and the algae grows as a biofilm. So it's a very dense biofilm on the porous wicking membrane. So the center, this is a cross section of a single synthetic leaf, and then if you look to the figure on the right, this is kind of how multiple synthetic leaves would fit into a synthetic tree system. So another advantage of this is that there is a lot of surface area for the algae biofilm in order to exchange gases with its environment, and there's also a lot of surface area for light delivery. Next slide, please. Great. A couple of applications for the synthetic tree. So on the left, we have a schematic of a scaled up system, or a drawing of a scaled up system. One application is bioproduct harvesting. So bioproducts are multiple. You can have energy positive biofuel generation. So unlike the open pond, we're not moving water around and we're not dewatering nearly as much. So it can be an energy positive production of these biofuel feedstocks. Solar powered production of high value products, pharmaceuticals or nutraceuticals that are produced by a particular alga of interest. And then also if you have a fish farm or something of that nature that needs food, this is an efficient way to cultivate feedstock for your farm. On the right, we have a concept of lightweight nutrient recycling, specifically for the synthetic tree gas exchange. So on a human space flight, this technology could be used to consume carbon dioxide and produce oxygen for a human crew, thereby acting as a gas exchange mechanism to close the loop on human life support in space. Next slide, please. Okay, so this is kind of getting into the details of how we design these systems. So at the top left here, our goal with these experiments was to figure out at what rate do you want to deliver, or do you want to passively pump or passively move nutrient media through this system, and that's governed by the evaporation rate from this terminal region. So in this picture on the top left, we have evaporation from this exposed region, and as water evaporates from that exposed region, fresh nutrient medium gets pulled in through the porous wicking membrane, and then the nutrients by diffusion feed the biofilm, and the biofilm consumes nutrients. So the plot at the lower left here has aerial bio. Oh, that didn't render quite right. I apologize. Aerial biomass density on the y-axis and time on the x-axis, and what we see here is what we expect, kind of an exponential growth of algae over time, but that exponential growth rate is higher closer to the nutrient medium reservoir. So as you go downstream, you're seeing a lower growth rate. You can see that in the pictures of the reactor on the if you look at reactor 1 at 20 days on the right side of the slide here, you can see that it's very green and dark close to the nutrient medium reservoir, but it kind of it looks dead or yellow farther downstream. And so the reason that's happening is because the algae cells closer to the or farther upstream are essentially getting the first crack at the nutrient medium, and take the nutrients out of it, thereby kind of depleting the nutrient medium of essential nutrients for their downstream neighbors. Next slide. And so how we mitigate this is essentially turning up the flow rate of nutrient media through the reactor. So on the left is kind of our first crack at designing this system. You have these small exposed regions, so you have a small evaporation rate and therefore a low flow rate of nutrient medium through the system. And if you look at the picture on the right, now we've increased the size of our exposed evaporation region by a factor of about 4 in order to increase the flow rate of nutrient medium through the reactor, similarly by a factor of about 4. And then if you see the inset on the lower right here, what we're seeing is we've mitigated the dead zones that are happening towards the top of the reactor in this case, in that the downstream, in this case upward, algae cells are getting ample nutrients from the nutrient media because the flow rate is faster through the reactor. Next slide. Okay, so this is kind of how we envision scale up of such a system. So we realized that the actual absolute magnitude of the height of the system is not what drives productivity. What drives productivity is this ratio of the height of one of the synthetic leaves to the spacing between the synthetic leaves. So these two schematics at the top, or these two cartoons at the top, are similar from a productivity perspective. They have equal algae surface area, and the one on the right is shorter and looks more like a synthetic lawn than a synthetic tree. So we kind of changed the goalposts a little bit through this to make it from a synthetic tree to a synthetic lawn, just because the synthetic lawn has the advantage of not having to passively transport water as high, which is more difficult because you're fighting with gravity in some applications. So on the bottom here, we see a scaled drawing of what a algae reactor would look like in order to completely consume all of the CO2 that is exhaled by a human, for example, for a human spaceflight mission. So for an 80 kilogram human, we're looking at a reactor mass of about 22 kilograms, which is significantly less. It's about 25x less mass, and that's mostly water mass than a conventional tubular photobioreactor for algae cultivation. So that's one of the huge advantages of this system, is that it requires a lot less water and therefore a lot less weight than conventional technologies. And based on the amount of CO2 that a human exhales and the amount of CO2 that algae inhales, we're looking at about a 4 meter by 2 meter by 10 centimeter rectangular prismic kind of shape to support one human in space. So I think if you go to the next slide, I think that's about all I have for this. And thanks for your attention. And I think we have a couple minutes for questions. So thanks, everyone.
Yeah, Tom, thanks. That was great. So yeah, we do have a couple questions. In the meantime, I'm going to stop sharing real quick. But the first question is, which cyanobacteria or algae have you tested with this technology?
Yeah, good question. We've tested a bunch. I think the one that we worked the most with was Anabina variabilis. And I think let's see, I think Lee's on the call. There are reasons that we chose that one, but I can't remember at the moment what they are. Chlorella vulgaris, we've also tried with this. That's a green algae. Anabina is a cyanobacteria. So we've tried a couple algae, like a couple true algae, a couple cyanobacteria, and it's like the common thread is they all seem to like it, kind of growing as a biofilm in this medium. One criteria is they have to have like some benthic capability in their kind of life cycle. So not all algae will work, but the ones we've tried have been generally successful.
That's great. And we have another question here. Would you be able to qualify relatively dry air and wet air in percentage?
Right. So percentage, we're talking about like generally relative humidity. It gets very close to 100% on the kind of inside of the reactor because there's just a lot of water in there. So the water, the gas phase and the liquid phase nearly equilibrate. So we're talking about like between 95% and 100% relative humidity on the inside of the reactor. And on the outside of the reactor, it just, in most of the experiments that we've run, it's just whatever it is. It's like it's a lab, so it's somewhere in California. It depends on the temperature and the weather, but typically between about 30 and 60% relative humidity.
Great. Thank you. Appreciate that. That was all very helpful. And so next up is Diana. Diana, can you see this? Or wait, let me share the screen. There you go.
Great, thank you very much. Can you guys hear me?
Yep.
All right, first slide, please.
Let's see, there you go.
All right, so biomaterials, and I'll just say up front that I could fill an entire talk with arguments over terminology for describing different categories and what does and doesn't count as a biomaterial, but for our purposes, we're going to define it very, very broadly as non-living materials made by life or incorporated into living systems. Have a lot of really interesting properties, and one that is pretty unique is that they tend to be, the structural materials tend to have a very deep hierarchy of structure. And what that means is So you take a traditional engineering material like, say, steel or iron, and a small piece of it is going to behave pretty much the same as the way a large piece of it is going to behave. And this is not true at all for most biomaterials. They have, for example, you might have a protein which forms into chains, and the chains form plates, and the plates are stacked together in kind of a brick and mortar arrangement to make larger plates, which are then distributed in such a way so that the macro scale piece that you get out the other side has extremely fine-tuned properties. It's light where it can be light, it's hard where it needs to be hard, it's strong where it needs to be strong. And that is a type of specialization that life is very good at making. The downside is traditionally it's very, very high overhead, meaning you generally need to grow a sheep to get wool, and you need to grow a tree to get wood, and so on and so forth. So the genesis of this project came about when we said, well, we know that these individual living cells already know how to do all of this micro and molecular scale environmental control and engineering that needs to happen to cause the small scale biomaterial formation to take place. Can we use 3D printed arrays of cells to control that formation process? So this is really a union of traditional genetic engineering with 3D printing used to control the cell physical environment. Next slide, please. So there are a number of parts that all have to work together for the proof of concept that we put together for this project. At kind of the upstream side, you need to basically develop a cell chassis. And so this is the cell that is going to make the material that you care about. You could implement this with a cell type, a cell line that just naturally produces the material that you want. But for our purposes, we wanted to show that we had a more flexible solution than that. So we used Saccharomyces cerevisiae, which is a standard model organism, basically baker's yeast. And that was constructed with a plasmid that allowed it to basically produce a fusion protein of our design that was tagged to secrete that material and to have a tag on it that would allow it to bind to the substrate. That cell line we then had to grow in a print medium, or you can think of it as being an ink, and that medium needed to support the cells in an inactive state prior to printing, because you don't want them to start reproducing or making the material that you care about before you've gotten them into place. Then there's the 3D printing process itself, where you need spatial control and positioning of the cells, and hopefully to not kill them while you are doing that. And then they're deposited onto a substrate, and the process of printing onto that substrate then needs to activate the cells. So hopefully keep them in relatively close to the positions in which they were deposited, but also activate them so they start making the biomaterial components that you care about. And then for our purposes, we also wanted to show that you could then remove the cells from the finished material. So you're left with essentially a non-living biomaterial construct. And so top to bottom, that's all of the components that we had to get working together. And can you go on to the next slide, please? And so here's a very short clip of the system in action. And can you play that video?
I did.
It's about 10 seconds?
Yeah.
Okay. Oh, well, you took my punchline away from me, but the point being that watching micron scale deposition and array construction, you can't really see a lot, but if you look directly under the dispensing nozzle, and this is just onto a plain glass slide for purposes of enhancing what you can visually see, we're making a 125 micron by 125 micron grid here. That's about 10 microns thick. And just to calibrate you, the yeast that we're working with are 5 to 10 microns in size. So this is essentially a single layer. So next slide, please. So the proof of concept, we're using 2 different carbon sources for our yeast to control the material production. And again, the proof of concept material that we're producing is a fluorescent tagged fusion protein. And so you can see that we have on the lower right there, you can see that we have suppression of material production prior to printing. We have material production after printing. We have pretty good coverage in terms of getting material deposited, and this is the upper right figure, in a test 25 grid array where we were trying to alternate between cells making green tagged and cells making red tagged protein. We had detectable material production in 24 out of 25 voxels, which is pretty decent. And so one of the benefits of this way of thinking about biomaterial production is that the material that you're making is not the feedstock that you're putting in, which is the way traditional 3D printing works. Instead, you're putting in basically feedstock for your cells, which is your basic, growth medium, sugars, etc., and you're producing a completely different material. You're not limited to bulk homogeneous materials. A lot of current microbial material production is on things like cellulose, for example, and this is getting at a different type of materials from that. But also, although you need some understand, if you want a cell to produce a material it doesn't produce naturally, and we picked yeast for our proof of concept because yeast are eukaryotes, and so the basic palette of materials they can make is, in a sense, broader and more likely to be useful than what you get out of prokaryotes. You don't need to fully understand the complete production pathway that the cells have, which is what you would need to do in traditional synthetic biology if you wanted to do this with genetic engineering alone, because you've also now got physical control over the environment that you're depositing the cells in. So next slide. So our original design goal was just to demonstrate the ability of this approach to produce a novel structural biomaterial, and we were thinking that this that advanced manufacturing was going to be the shortest term application for this. But as we've invested in it a little bit more, we're also starting to see some other people who are interested in using an approach like this for reproduction of 3D structured cell cultures, so biofilms, microbial mats, and actually bio-enabled filters. I was interested to see the algae system in the previous talk because it's similar to an application that we've also discussed for something like this, where you could be creating a filter with algae embedded in it as a single piece, and also enabling research into cell response to microenvironments in 3D, which is a very big thing, particularly for understanding life in extreme environments. Cells there are extremophiles are frequently not experiencing the stresses measured in the bulk material, but rather they're limited to and controlling more favorable microenvironments. And so I believe, is that my last slide? I believe that's my last slide.
Yes, it is.
Yes, it is. All right. So with that, I'll take questions.
Yeah. So as people put the question in the chat, is this something that can be scalable? Like, does it have a limit? I remember John saying in the beginning, but it could be some completely different. Where it seems like with biology, you can kind of scale it pretty big. Can that be the same? Can the same be said with this?
So if you're basically creating if the array that you are doing the 3D printing of is then something that is going to grow into a larger framework, there's essentially no limit to the size that you could be dealing with. if you're just, as we were doing in the proof of concept, if you're just using the cells basically as a temporary means of local material production, because your end goal is something that is non-living, then you're going to be limited to the scale of whatever your 3D printer is. And of course, there's other research in that area on scaling up 3D printing and using kind of a extrusion printing that doesn't have a length limit and so on and so forth.
Oh, very interesting. It doesn't seem like there's any other questions. So yeah, thank you for presenting. And if there's any questions that kind of filter in, we can send them your way to get them answered. So I appreciate you presenting. It was very insightful.
All right, thanks. Take care.
Yeah, take care. All right, everybody, just give me 1 second here. All right, and our next presenter is Jing Li. Jing, take it away.
Knowledge. This is an issued US patent. The nanosensor array can be used to form an electronic device to mimic human nose, then to smell or detect odors or chemicals. Each sensor in the array functions as a node inside the human nose. So this device with a nanosensor array is also called NASA electronic nose technology. So the odors from human breath can be correlated with different medical conditions. Next slide, please. Yeah, so Excel breath is a window into your health. So there's a different size of the molecules from the Excel breath. The small molecules like nitric oxide, ammonia, and carbon dioxide can be biomarkers for certain medical conditions. Then the volatile organic compounds, we also call the VOCs, like acetone, alkane, ketones, and benzene, they can be also the biomarkers for to correlate with the different disease. And the larger and non-volatile molecules like a protein contained in aerosol, those proteins can be contained in the aerosol droplets. And here, with the nanosensor array, we focus on the volatiles, which means the sensor array detect the small molecules and the volatile organic compounds. So the NASA eNose device on the lower right corner, you can see is the first version of the eNose uses an array of 32 different sensors to mimic a human nose to smell the Excel breath for medical diagnosis. Next slide, please. So we have developed two types, or say two versions of eNose devices. Both are designed for one-button push operation. The left images show the first generation of the eNose, as you have seen in my previous slide. So this device we have developed around 2012 with a plug-in to an iPhone and controlled by the app on the phone for trace toxic chemical detection. The sensor data can be displayed on the phone and transmitted to or stored on another phone or a computer or on a cloud. For whomever interested in the data, they can download the data from the cloud. So this effort was funded by Department of Homeland Security with about $4 million. And now we extended the eNose technology for medical applications and started to work on it with the Lawrence Livermore National Lab and the Travis Air Force. So the left side is the press release from Travis Air Force. And okay, so the right images showed our second generation of the eNose device. that is controlled by an app on the small iPad mini via Bluetooth. So now this eNose device is not directly connected to the smart device. It be communicate or controlled by the smart device via Bluetooth. This is currently funded by Department of Human and Health Service with the $3.8 million to enhance NASA eNose device, specifically to tune the nanosensor array inside the device according to the breath profile obtained by the GCMS analysis for COVID-19 detection. So the right side is the recent press release from NASA. Next slide, please. Yeah, so this slide, we are going to talk about the heritage of the eNose technology. We have worked on this technology since 2001. We built an initial eNose type of device and launched it to space in 2007 on the Navy satellite for trace chemical detection. And then this is the first ever nanosensor array device flown into space. And the second flight opportunity was we put two of our nanosensors on JPL eNose platform deployed on ISS for environmental monitoring of chemicals such as alcohols, ammonia, formaldehyde, sulfur compounds, and mercury in 2009. Next slide, please. Yeah, on this slide, I'm going to tell you the working principle of the eNose technology. So the eNose device uses an array of sensors, not just single sensor. You can see in the middle lower part image. So the sensor array will measure, will expose to the chemicals. And so we are measuring the change of resistance when chemical molecules attach to the surface of the sensing material. You can see in the, Jay, would you please click one more time? Yes. So you can see the left side upper image shows how the gas molecules or the organic volatiles molecules come in contact with the carbon nanotubes, one type of nanosensors in the array. And so when it's attached to the surface of the carbon nanotubes, it will have the charge transfer interaction occurred. And that charge transfer interaction can cause the conductivity change of the materials. Therefore, we measure the resistance change as our sensor signal. Since we use array of sensor, so we will, when the sensor array exposed to a chemical, we will get the sensor response from all 32 different sensors. So then we plot the sensor response into a bar chart. Then we can obtain a pattern, like the bar chart as a pattern. And then we can use the pattern recognition algorithm to do the classification, as it's shown on the right side. And this is we're using the support vector machine learning algorithm to do the classification. And in this plot, you can see the green color is the air, and the red color is the chlorine, and the black is the ammonia. So if the measurement data fall into the black area, that will be identified as ammonia. If the measurement data fall into the red area, that will be called chlorine. So that's how we do the identification. And the middle upper part is the sensor response to different concentrations of CO2. Next slide, please. So here we show that sensor response to nitric oxide measured by expensive and desktop instruments on the left side, and then the same concentration of nitric oxide measured by our eNOS device. And so you can see that we miniaturize our electronics to make the sensor array device that almost show similar performance as you can see in terms of detecting parts per billion concentration of nitric oxide. So this is the medical relevant concentration range. And nitric oxide is a key biomarker of a lung injury that is important for NASA and for military and the civilian as an example. Next slide, please. So in summary of this invention for medical application, using breath analysis and the eNOS with an nanosensor array, it can provide the capability as non-invasive diagnostics and monitoring health status and fitness of personnel on long space missions and also in the civil life. It can detect emerging infections and internal diseases in early stage. And it can be quickly and on-the-spot screening using this sensor array device by a smartphone or smart tablet. So the challenges to be addressed with for this technology is we need to find biomarkers for relevant medical conditions. And we need to tune the sensors for specific biomarkers to be sensitive and specific. Sensor signal processing and machine learning models need to be tuned specifically for certain medical conditions. And the impact of this technology is eNOS can provide non-intrusive, real-time, mobile, and autonomous monitoring of health status. It can provide early warning for abnormal physiological condition and diseases. So healthcare can be performed at the point of care or at home. And this technology also provides the possibility to do the telemedicine. So that is all my presentation. Thank you for listening.
Thank you, Jing. That was fantastic. Appreciate that. We do have a few questions. The first one is, what is the patient number for this technology? I'm assuming that means how many people has this been used on and tested?
We just started. Started to tune the sensor for COVID, and the target number of patients will be around 200 patients, but we have not started yet, and we are in the process of getting the IRB approved so we can take the human subjects as the test subjects. So yeah.
Great. Thank you.
Okay.
Few more. What is the accuracy of detection? Do you have any data on FAR, so false acceptance ratio, and FRR, so false rejection ratio?
Not on the humans, because we have not tested this device to the humans. We are still in the process, as I just said, getting the IRB in place so we can move on to the human subjects. From the lab, and we have tested the chemicals in the lab, and we have 72 samples and using the classification algorithm to identify those or say cross-validate the identification of 72, and 1 out of 72 was further away with some uncertainties, and 71 were within 95 certainty of identification.
Oh, got it. Cool. And we have two more questions. I guess I can just pose both of these at the same time. Is the sensor array self-cleaning, or does it need to be replaced frequently? And where do you see the use of eNOS technology other than medical, so beyond that?
Okay. The first question, it depends on the application. And so far, we are tuning our sensors to the biomarkers of the COVID-19. And from our lab testing to those VOCs, and we saw our sensor can recover by just a flush with the room air. And if we encounter very sticky chemicals or VOCs, we have a way to remove that chemicals by UV illumination. So the UV light can break the bonds and release the chemicals from the connection to the sensing material or to the surface of sensing material. Yeah, that's the way how we can refresh the sensor surface. Next question is, other than medical applications, we see this can be used for like food industry. And it can also be used in the chemical industry, like incoming raw material detection. So currently, I know when the raw materials come to the chemical plant, like a toluene or acetone, they need to stop in the gate and have someone climb the ladder to the top and scoop the chemical and then send it to the analytical lab to run the detection. So if we have the eNOS, which is mobile and easy to operate, so we can do the detection at the spot. So when the truck come in, so we can detect the incoming materials at the spot. That will be quick and doesn't have to go to the lab. Another one is like a production line. So for the industry, like people produce the candies with the strawberry flavor versus the cherry flavor. You know, visually they are very similar, but when you smell, they are different. So when the production switch from one candy to another candy type, so right now they use human panel or human nose to smell, but human nose can be tired in 20 minutes. And so if we use the eNose, that can be easily do, and it will be even more objective.
Oh, wow, that is very interesting. And then, so we do have one final question here. Would it be more accurate for bacterial infections rather than viral ones, if that's been studied already?
We don't know which one will be more accurate. Depends on which method will produce more distinct biomarkers in the vapor phase. I don't have data for one or another. I think that will be an interesting study for us to look into it.
Yeah, yeah. Well, great. Thank you, Jing. That was fantastic. Appreciate it.
Yeah, thank you for all the questions. Thank you, Jay, for organizing this.
And next up is David. David, take it away.
Good morning, everybody. So I'm excited to be able to tell you about some medical applications of nanotechnology. And before I dive into the slides, I just want to make a comment. I'm one of the few physicians who works at NASA Ames Research Center. And NASA Ames Research Center really does a lot of interesting technology development. A lot of it comes from outside traditional biomedical disciplines, comes from material science, nanotechnology, other physical sciences, and it's really an awesome place to do medical technology development because you can borrow from these fields that are outside of sort of the mainstream medical world, and you can bring those technologies to a medical arena, and that's a very exciting thing. So I feel like a kid in a candy store getting to do this at NASA Ames. So I just wanted to make that comment. Next slide, please. So since I'm going to talk to you about two different technologies, both based on carbon nanotubes, I'll just give you a brief overview of what carbon nanotubes are. This is a new form of carbon that was originally discovered in the early 1990s. Hadn't been known before. It can be thought of as a two-dimensional graphene sheet rolled into a tube. It can exist in a couple of different major forms, single-walled carbon nanotubes, multi-walled carbon nanotubes, and other forms. And it has interesting electrical properties that can be almost metallic in nature from the standpoint of conductivity, or it can be semiconducting, which really opens up a variety of different electronic applications. Carbon nanotubes also have pretty amazing mechanical properties. They're very robust and very strong, and it's ideal for a number of material science applications, but also ideal for a number of medical applications where that strength and durability is important. Next slide. So carbon nanotubes have really amazing mechanical properties, amazing electrical properties and chemical properties. But the one property that I really want to highlight is their biocompatibility. From the standpoint of a physician trying to look for medical applications of nanotechnology, the biocompatibility is really important. Carbon nanotubes do not trigger an immune response, is really what's quite exciting about them. There were some initial reports that carbon nanotubes, in fact, showed some toxicity, showed some inflammatory responses. But when you get down to preparations of these materials that are very pure and free of contaminations, you can really demonstrate how biocompatible these materials are. And in this picture at the bottom of the slide, I'm showing a carbon nanotube-based material that's implanted into the retina, and it shows excellent biocompatibility. If this had been a non-biocompatible material, you would have seen a big halo of inflammation and edema and swelling at the implant site, but that was not seen. So this is a very nice demonstration of just how biocompatible these materials are. Next slide, please. So this first project using carbon nanotubes is a retinal light processing technology. And basically, it's the development of an implantable device consisting of an array of carbon nanotubes on a silicon chip for the purpose of restoring vision in patients with macular degeneration and other types of retinal disorders. So what is macular degeneration? Next slide, please. So macular degeneration, it's often called age-related macular degeneration, is actually one of the most common forms of blindness in the world. Next slide, please. And it's a disease that's characterized by loss of central vision due to death of the retinal pigment epithelium, or death of the photoreceptor cells in the retina. Next slide, please. So this is a picture in both a schematic form and in a tissue hematoxylin and eosin preparation on the right, showing the various layers of the retina. So it's a multi-layered structure, a highly organized architecture, as you can see. And what's interesting about macular degeneration is that this lower layer down at the bottom, called the retinal pigment epithelium, is what's damaged in patients with macular degeneration. All of the remaining portions of the retina remain intact, but if you damage the retinal pigment epithelium and then you damage the adjacent photoreceptors, you cannot receive optical signals from the outside world, and you cannot transmit the electrical impulses that are generated from those optical signals to the brain, and you cannot see. Essentially, you're blind. Next slide, please. So the whited out portion of the tissue here is the section of the tissue that's damaged in degeneration. But the remaining layers of the retina actually are very well preserved in patients who have this ocular disease. And that's a very tantalizing fact. Next slide, please. That's a very tantalizing fact, because if you can somehow bridge from the outside world some sort of optical signal to the intact layers of the retina, these upper layer portions of the retina where the nerves are fully intact, if you can bridge that damaged area with some sort of device that spans that physical distance, you have the potential to restore vision in these patients. So what you're seeing here is a very simple schematic diagram of a silicon chip depicted in orange, with a single tower or a single electrode made of carbon nanotubes that spans that distance through the retina and potentially delivers optical signals where they need to be to help restore vision in patients with macular degeneration. Next slide, please. So the concept behind this technology is to provide some sort of an optical signal, some sort of a two-dimensional optical signal through some sort of external camera, in this case depicted as being mounted on the lens of a patient's spectacles, their glasses, and then to take that signal and to transmit it to an array of carbon nanotube towers, an array of carbon nanotube electrodes that are grown onto the surface of a silicon chip. This silicon chip, shown with many different carbon nanotube towers on it, in orange, depicted in orange, would then be implanted into the back of the retina and pressed into place. So each one of those electrodes would penetrate through the retina to the active neural layer, the undamaged neural layer of the eye of the retina. So it could deliver optical signals that would be generated by some sort of external device. And in this particular picture, I'm showing this as a hardwired technology, but in reality, it's not necessarily that this be hardwired. It's merely necessary that you implant the chip into the back of the retina, and then you can use all kinds of short distance wireless technologies to deliver that technology from an external camera mounted on the glasses to the chip that's implanted in the patient's eye. Next slide, please. So the technology that NASA Ames has developed is not the only technology of this type. In fact, there are a number of different companies that have developed similar technology. One of the most successful companies is a company called Second Sight that has developed a system that they call Argus. They now have Argus 2, and a patient who's outfitted with this device wears a special set of glasses for acquiring images, and then those glasses are coupled to an external coil, device for transmitting those signals to the chip that gets implanted into the patient's eye. And then that chip is able to successfully deliver signals to the retina. And then those signals are transmitted to the visual cortex of the brain. So a patient can actually begin to see shapes, can see light and dark, and can begin to have some replacement of the vision that they've lost from a disease like macular degeneration. The number of electrodes that are currently used in a technology like this is almost 400 individual electrodes. So it's beginning to give some spatial resolution of images to the patient. It's quite short of full vision, but for the patients who have macular degeneration and other related diseases, it allows patients to move around their homes with ease and to recognize family members, to go outside and to have a much expanded range of activities than they could have before, prior to this type of technology being developed. So it's a big win for the patients who have these very debilitating forms of blindness that it's able to treat. Next slide, please. So I'm going to move into this second technology, and I'll ask your patients on questions. We'll have all of the questions for the entire presentation at the end, if I can ask for your patience on that. So I want to talk to you about a second carbon nanotube based medical application that involves using a device that we call the NASA biocapsule, basically as a drug delivery technology for quite a wide range of applications. Next slide, please. So I've talked to you about carbon nanotubes, and I've shown you some examples of how carbon nanotubes can be fashioned into electrodes that can penetrate tissue. But I want to shift gears now and talk to you about a very different form of carbon nanotubes that we call bucket paper, which is essentially a meshwork of carbon nanotubes that are formed into a flat paper-like structure. You know, it's kind of ironic that we're talking about nanotechnology, but as you can see in this picture, the person holding the forceps is actually holding a piece of bucket paper that's quite big. This is about a 1 centimeter piece of bucket paper. So nanotechnology implies really small things, and certainly the material properties of carbon nanotubes are based upon their ultra small structure. But even nanotechnology can have applications that are very macroscopic in nature, and here's a classic example of that. So we can make a carbon nanotube meshwork, but for a variety of medical applications, we would love to have a 3D structure, not just a flat structure, a paper-like structure, but a 3D structure that we can actually form into a container. Next slide, please. So here's a low-resolution scanning electron microscope image of carbon nanotube bucket paper. It's a smooth surface at this relatively low resolution. But the next slide shows, the next slide, please, shows that this meshwork of carbon nanotube bucket paper is really a structure that has pores to it. It has passageways through which gas molecules, water molecules can pass. So if one can think of creating this into a three-dimensional container, one can really take advantage of the ultrastructure of this paper-like material. And that opens up a whole broad range of medical applications. Next slide, please. So what we have developed at NASA Ames is a way of making bucket paper into these hollow containers that I have alluded to a couple of slides ago. And it's a wonderful method. It's a very simple method that just involves taking a mold that is perforated on its surface, as shown in panel A, for instance. You can see the blue dots represent perforations of some physical mold that might be made of steel or some other inert material. And then by applying a vacuum to the inside of this mold, one can draw a solution containing carbon nanotubes to the surface of this mold, and one can draw the solvent into the interior of the mold, and over time, eventually achieve a complete sort of matted structure on the surface of this perforated mold that allows you to form a nice three-dimensional capsule, or in this case, really a half capsule that can then be used to form a complete capsule. Next slide, please. So if one forms this mold, forms this half capsule, if you will, on the mold, one can then slip the mold out of the half capsule. Next slide. And now one can simply take these two half capsules, join them together, much the way the halves of a medicine capsule are joined. And now where it really gets exciting is you can put some cells inside of these capsules. And the green structures with the pink nuclei are designed to represent cells that can go inside the capsule. Next slide, please. So to give you a sense for where we're going with this technology, this single figure really describes how we're using these capsules to contain cells. For this particular application, we're talking about placing islet cells into the capsule. Islet cells are the cells that produce insulin in the pancreas. So they have both a glucose sensing function and an insulin secretory function. And if you place these islet cells into the capsule, you can take advantage of the fact that the capsules are very biocompatible, and you can now implant this capsule containing the islet cells into a patient with diabetes, so that you can now replace their islet cells, which are lost as a result of diabetes, and you can essentially give back the islet cell function that they lost with the disease diabetes, and you can restore normal glucose sensing and insulin secretory function in a very effective way that has the potential to cure them of diabetes. The biocompatible capsules protect the cells from attack by the host immune system, in particular by immune system cells such as killer T cells, shown in pink in the upper left-hand portion of this figure and the lower right-hand portion of this figure. It can, the capsule, because of the pore size, does not allow those immune system cells to enter the interior of the capsule. But at the same time, the pores allow the free movement of carbon dioxide and oxygen across the capsule wall to keep the cells inside, to keep the islet cells inside happy and healthy, and keep them at the physiological temperature where they need to be in order to function in the normal physiologic way. So this system allows the islet cells inside to function as they would in the pancreas. Next slide, please. So I'm going to summarize what I've talked about in the following way, say that really because the carbon nanotubes exhibit such a high degree of biocompatibility, they really lend themselves to a wide variety of medical applications that involve implantation into the body. And that can be in the form of electrode type physiological implantations, taking advantage not only of the biocompatibility, but also their electrical properties, but also can take the form of a larger material, more of a three-dimensional material for encapsulation of living cells for drug delivery purposes, really for a wide variety of drug delivery purposes, only one of which we've touched on today. Treatment of diabetes is the holy grail for this type of application, but I think there are many other applications that are possible, such as the delivery of cancer chemotherapy agents in a very localized way, for instance, into a tumor bed that may be inoperable, and also implantation of cells for gene therapy applications, again, where you want to have perhaps a localized delivery of those gene products as opposed to systemic delivery of gene products, although both localized and systemic delivery options would certainly be possible. So thank you very much for your attention, and I'll be happy to answer questions on both technologies, the retinal processing chip and the implantation technology for cells.
Yeah, thank you, David. That was very informative. Thank you for going over those two technologies. So we do have some questions that are getting lined up. First one is, do you see any possibilities of using carbon nanotubes to reduce or eliminate macular blisters in a patient by lessening the inflammation that caused the macular blisters? Halting the macular issues before they become worse seems to be a key approach.
That's not an application space that we have investigated, but I will just say very generally that if carbon nanotubes could be used in a physical sense to prevent or reduce blister formation, there might be a possibility there. It's also possible that some sort of carbon nanotube-based drug delivery system that would allow very long-term sustained drug delivery could potentially have an impact on that type of disease process. But that's speculation on my part.
And the next question is, how would carbon nanotubes fare in space microgravity environment?
Yeah, carbon nanotubes, really like any molecular form of carbon, should be perfectly fine in space. I wouldn't expect any effect of the space environment on the carbon nanotubes per se. How the space environment will affect some of the medical applications more specifically is yet to be seen, but certainly the carbon nanotubes themselves should be fine.
Great. And another question here is, yeast scale up to more than 500-liter runs into major issues, right?
I'm not sure that question
would that be
relates to this. Was this perhaps a question related to another presentation?
It might have been. Yeah.
Okay.
Yeah. Sorry about that.
That's all right.
Does this apply to both dry and wet age-related macular degeneration?
Yes. It really does. The more severe forms of macular degeneration, the wet macular degeneration with the neovascularization process that takes place is really more of the target application, because in those cases, patients have lost essentially all of their vision, whereas the dry form is a less severe form of the disease, and usually there's some vision preserved in those patients.
Okay, yeah. And the next question is there a time limit of a person having macular degeneration issues that will not work, but then they implant to increase the sight?
That's a great question. I think the current understanding is that the intact neural layers within the eye remain intact and remain accessible to this type of intervention for a long time. So I don't know of any time limit for intervening with an implantable retinal chip.
Okay. And this seems to be the last question, unless some others trickle in. For encapsulated cell implants, are there risks of the cells secreting antigens through the Bucky paper? Is it possible to cause persistent immune responses?
That's an excellent question, and the answer is yes. There's certainly a theoretical concern that just because the cells stay inside the capsule doesn't mean that some antigens that are derived from those cells might not be released by the cells, get across the capsule wall, and enter the general circulation within the body. The only comment that I will make about that notion is that the ability to put really any cell type within the capsule, we feel, is such an advantage. One can choose engineered cells, for instance, that have low antigenicity, that are less likely to release antigens. You really have a lot of flexibility and a lot of options in terms of the type of cell you put in. So you can certainly choose cells that are less likely or perhaps not even capable of releasing antigens that could leak outside the capsule. But that's an excellent question.
Okay, great. I think that's the end of the questions. So thank you, David. I appreciate it.
Thank you very much. Thanks for the opportunity to speak.
Yeah, it was great. And if we can get a virtual round of applause to all the innovators who were able to speak. Everything was so fantastic presentations and it's very informative. So next up, I'm going to quickly go through how to leverage all this NASA technology to benefit your company. We heard these innovators talk about all of the amazing things they've come up with, and now it's the technology transfer's office job to show you how you can come and license the technology. So kind of a quick broad overview of the NASA Technology Transfer Program. I mentioned earlier in the beginning of the webinar that we have over 1,000 patented technologies, and those come from the 10 different NASA centers. And we're here, as you can see in the green, in Silicon Valley at NASA Ames Research Center. But all the rest of the centers are spread all throughout the country. So the NASA Technology Transfer Program, since its inception over 50 years ago, the main goal of the program has been to ensure that innovations developed for exploration and discovery are broadly available to the public by maximizing the benefit to the nation. And the question always comes, why should a company license NASA technology? A NASA license allows access to a technology for testing and to implement it into a system, a service, or a product that could result in sales for your company. There are many potential quantitative measures too, and since our spin-off publication 2000, NASA is able to quantify these benefits with over 18,000 jobs created, $5.2 billion revenue generated, 18.6 billion in productivity and efficiency. So that means costs avoided in total by companies who provided that data. 30 million lives improved, with the majority of that coming from unique nutritional supplements used in baby formula and new materials used in surgical implants. And then, of course, the environmental impact. Space-based technologies can help with remote sensing, have enhanced scientific understanding of water cycles, air quality, forests, agriculture, and things like that. And I did just mention the spin-off publication, and that is our publication that showcases all of the NASA licensed success stories, and I'll share that link at the end of the presentation. So if you want to license a NASA technology, you'd have to go to our website, which is technology.nasa.gov, and this is the page that comes up when you get to that website. So there's two ways to search for NASA technology. First way is putting in keywords in that search bar where it says search for NASA technologies, and the other way is clicking on the NASA patent portfolio. So we'll be going through that route. Once you click that, you'll see there are 15 different categories of licensable technologies, and they range from aeronautics to manufacturing, propulsion, and like today, health, medicine, and biotechnology, environment, sensors. There's just a whole slew of technologies where you can come check them out, see if they fit what you want to do, and you can apply for a license. And since we went over the health, medicine, and biotechnology category, we'll go over that and show how it looks if you want to apply for a license. So once you click the health, medicine, and biotechnology category, a page like this shows up. And this shows the different technologies that are available for licensing. And so let's go ahead and click on the biochemical sensors using carbon nanotube array. And once you click that, you see this page. And this page gives you an overview of the technology with understanding what the technology is, the benefits, the applications, and the technology itself. And if you have any questions, you can click on the contact us link that's highlighted right now. And then if you, let's say you want to take it on the road, you can download a PDF of the technology, of the information. And the PDF looks like this, which is exactly the same stuff that you saw on the website, but it gives you a PDF so you can take it offline. Another important part is the patent. So majority of our technologies will have the patent link available. So as you can see, it has the patent highlighted right here. And once you click on the patent, it'll take you to the USPTO office. And this is where it's very important. After reading the patent, make sure that you go over the claims and the description of the technology so you know if it meets your needs. And so I'm going to give you a quick run through of how to apply for a license. So as I mentioned before, you can apply for a license on that main landing page of the technology. So when you click on the page, you can then see the apply now to license this technology. And you can also learn more about how the licensing structures, the fees, and everything like that at technology.nasa.gov/license. And one very important note here is that you must be illegal. Your company must be legally formed in order to apply for a license. So after you click on the apply now, you come to our Atlas page, which stands for Automated Technology Licensing Application System. You would click on the create guest account, and once your account is created, the license application page should open. And the first step is selecting the licensing type, and I'm going to be going over the 3 types. So you have the 3 types of licensing agreements: a startup, an evaluation, and a commercial license. And so with the startup license, it's a non-exclusive license, which means companies may apply for similar rights to use the technology for commercial purposes. And companies entering this license agreement are bound by all requirements in federal licensing, just like all of the other licenses are. And while NASA does license to foreign entities, this startup agreement is only available to companies in the United States. And another interesting little tidbit about the startup licenses is that NASA waives the initial licensing fees, and there are no minimum fees for the first 3 years. The next type of license is the evaluation license. An evaluation license are non-exclusive agreements without any long-term commitment. These licenses range from 6 to 18 months, and most licenses are around a year. The evaluation licensees are not permitted to commercialize or sell the technology. So to do that, you would need a commercial license. So pretty much you can think of it as kind of a test run license where you see if the technology would fit what you want to do. And then the commercial license is your standard commercial license, and it's available for domestic and international organizations. And the terms, just like with the other licenses, the terms are negotiated on a case-by-case basis. So those are the 3 licensing agreement types. So applying for the, after you apply for it, you click on the type of license you want. The last few questions, the first section, so you can see right here, of the application will determine your license type. So startup, evaluation, commercial. Before you advance the next section, check at the bottom of the page to confirm that your intended license type is listed. To complete the rest of the application with as much detail as possible. And then after that, click submit on the last page, and to be sure to complete the Adobe e-signature process. Once you've signed once you've e-signed the PDF correctly, your application is submitted, and you'll receive a confirmation email that your application has been submitted. And again, it's very important that you fill out the application with as much detail as possible, because then that will help our licensing managers with your application. So the more, the better in this case. And I want to touch on our NASA software catalog. So we have free software available at software.nasa.gov, and we have 3 different release types. And this is open to companies and individuals. So if you're somebody who's somewhat of a hobbyist and you want to see if there's something out there that might help one of your hobbies, go ahead and apply for it. And the 3 release types are laid out in more detail on the website. So you have your government purpose. So for that, you need a government contract. General purpose, which has 3 different categories: U.S. only, so you have to be in the United States; U.S. and foreign, which is open to U.S. and foreign people in foreign countries; and then public. And we also have our open source technologies, which is at code.nasa.gov. And the little asterisk, as you can see here, so all but open source software requires software usage agreement. So there is an agreement involved with all of the software other than the open source software. And these are some helpful links on the program, how to license NASA technologies, the NASA patents, free software. We've also included SBIR, STTR, which are small business innovation research. Success stories, which is when companies license our technologies and they take it and they create very, very cool things that are out there in the real world. And then NASA Home and City, which is kind of like a virtual way to, you can see which things in your room or around the city have been through NASA, that have been spun off through NASA. And my contact information is below. And one last question that I want to leave you with is, what is left undone in your company or in your project that you might search NASA technology for a solution? So thank you everyone for attending this webinar. I hope it was informative as it was for me, and we look forward to seeing you at our next one. Thank you.